Welcome back to the Deep Dive. We're here again to really get into some fascinating research, pulling out the key stuff just for you.
That's right.
And today, we're looking at something, well, really common. Might be in your kitchen right now, actually, egg white.
Albumin, technically.
Exactly, albumin. But we're definitely not talking breakfast here. We're digging into its surprisingly potent scientific side, especially for healing and tissue repair.
Yeah, the sources we have today are, well, they're pretty compelling. They cover quite a spectrum, from almost traditional approaches for burns,
Right.
all the way to really cutting-edge stuff, biomedical engineering, 3D printing.
And even skin aesthetics, pigmentation issues during recovery.
Exactly. It's a really interesting spread.
So our mission today is to unpack these studies. We want to really understand what researchers found out about egg white and, how this staple ingredient could actually be important for wound healing in ways you probably haven't thought about.
It's going to be quite a journey.
It is. We'll start with a clinical trial on burn healing, then move to an advanced, 3D printed dressing involving stem cells.
And then dive into how egg white components might affect skin color changes after injury.
It really shows how science can take something familiar and find layers of complexity.
It really does. Often the simplest things hold surprising secrets.
Okay, let's get into this first source then. It's a clinical trial published in the Avicenna Journal of Phytomedicine, and this one investigated an ointment made with egg white for patients with second-degree burns.
Yeah, and burn wounds, we should probably just emphasize up front, they're a massive global health issue.
Really?
Oh, absolutely. They rank as the fourth most common type of trauma worldwide. After things like car crashes, falls, interpersonal violence, burns are right up there.
Wow, fourth. I wouldn't have guessed that high.
It's significant. And the impact isn't just the initial injury, it's the complications, infections, terrible scarring, long-term disability. So finding treatments that are effective, safe, maybe even cheaper, that's a huge priority.
And this study focused specifically on second-degree burns.
That's right. Partial thickness burns. These damage the top layer of skin, the epidermis, and part of the layer beneath, the dermis.
Okay.
They hurt a lot, they can get infected easily, but the key thing is they can usually heal on their own, without skin grafts, if they're managed well.
So a good place to test something aimed at speeding up that natural healing process.
Exactly. It's a good model for treatments promoting skin regeneration.
All right, so tell us about the study itself. How did they set it up?
So this was done back in 2017 at Taleghani Hospital in Avaz, Iran. And importantly, they used a triple-blind, randomized clinical trial design.
Triple-blind. Okay, that sounds rigorous. Remind us what that means.
It's definitely a strong design. Triple-blind means 3 key groups involved in the study are kept in the dark about who's getting which treatment. So that's the patients themselves. They don't know if they're getting the real deal or a placebo. The clinicians or researchers who apply the treatments and assess the wounds, they don't know either. And finally, the statisticians or data analysts crunching the numbers, they're blinded too.
Ah, okay. Patients, clinicians, analysts. Why go to all that trouble? Why is blinding so important?
It all comes down to minimizing bias. Bias can creep in so easily. A patient might feel like they're healing better if they think they got the new treatment.
Right, the placebo effect?
Exactly.
Yeah.
Or a researcher might, even subconsciously, score a wound slightly better if they know it's in the treatment group they're hoping will succeed. And analysts might handle borderline data differently. Blinding everyone involved means any difference you see between the groups is much more likely to be genuinely due to the treatment itself and not just expectations or subtle influences.
Makes total sense. It really strengthens the evidence. So who were the patients in this trial?
They recruited 90 patients, and they had pretty specific criteria. They had to be adults, aged 18 to 65.
Controls for age-related healing differences.
Right. And the burn couldn't be too extensive, less than 15% of their total body surface area. Larger burns are just a different ballgame, needing more intensive care.
Got it.
They also needed patients who got to the hospital relatively quickly, within 6 hours of the burn happening. That helps standardize the starting point, the initial condition of the wound.
Okay. Any reasons someone might be excluded?
Oh, yes. Quite a few important ones. Burns on sensitive areas, face, hands, genitals, perineum were out because those areas heal differently and have, major functional or cosmetic importance.
Sure.
Also, patients with underlying health issues that could mess with healing were excluded. Things like diabetes, immune deficiencies, active infections elsewhere,
Right.
and people taking certain medications known to affect the immune system or healing, like steroids or immunosuppressants. Oh, and crucially, anyone with a known allergy to eggs. They did a patch test first to make sure.
So they really tried to get a group of patients who were, generally speaking, healthy, with similar types of recent blends in less complicated locations.
Exactly. The goal is to isolate the effect of the treatment as much as possible by making the groups comparable in every other way. It boosts what we call internal validity.
Okay, so they had their 90 comparable patients. How did the actual treatment work? What did they apply to the wounds?
All right, so the 90 patients were randomly split into two groups, intervention and control. Now, importantly, both groups got the standard treatment for second-degree burns used at that hospital,
Which was what?
which was applying a 1% silver sulfadiazine cream. It's a very common topical antibiotic for burns, mainly to prevent infection.
Okay, so everyone got the silver cream. What was the difference then?
The difference was what went on before the silver cream. The intervention group got the egg white formulation. It was prepared in an ointment base, applied directly to the cleaned wound. Then the silver sulfadiazine went on top.
And the control group?
The control group got a placebo ointment applied first. This placebo looked and felt like the egg white ointment, but, without the actual egg white. Then they got the standard silver sulfadiazine cream on top.
Ah, okay. So the egg white or placebo was an add-on to the standard care, not a replacement.
Precisely. Then both groups had their wounds dressed with standard dressings, changed daily or more often if needed.
And how did they track the healing progress? How did they measure if the egg white was making a difference?
They used a validated assessment tool called the Bates-Jensen Wound Assessment Tool, or BWAT. It's quite comprehensive.
What?
It has 15 items that look at different characteristics of the wound and the skin around it. 13 of those items are scored on a scale from 1 to 5.
And 1 is good, 5 is
Right,
bad.
exactly. A lower score means better healing, healthier tissue. So a decreasing total score over time is what you want to see.
Makes sense.
They perform these BWAT assessments on day 1, the baseline, then again on day 7, and finally on day 15. They also measured the wound size, just length and width, at the same time points.
Okay, now we get to the really interesting part. The results. What happened with those Bates-Jensen scores? Did the egg white make a difference?
It really did. This is where the findings were quite dramatic. The average BWAT scores dropped significantly faster and lower in the group getting the egg white ointment.
How much lower?
By day 15, the average score in the egg white group was down to about 13.75 plus or minus 1.83. In the control group, the average score was still much higher, around 21.51 plus or minus 5.7.
Wow, 13.75 versus 21.51. That's a big gap. Was it statistically significant?
Highly significant. The paper reported a p-value of less than 0.001 for that difference on day 15. That means there's a less than 1 in 1,000 chance that difference happened just by random luck.
Okay, p less than 0.001. That's really strong evidence. What did that mean in practical terms? Like, how fast did the wounds actually heal?
This is maybe the most striking part for, someone experiencing a burn. The study found a major difference in healing duration. Most patients in the egg white group showed significant improvement, basically reaching near complete healing within just 5 to 10 days.
5 to 10 days. That's quick for a second-degree burn.
It is. Compare that to the control group, where most patients took considerably longer, typically between 10 and 17 days, to reach a similar point.
So you're potentially cutting the recovery time by, what, a week? Maybe even more for some people.
That's what it suggests. And imagine what that means. Less pain, lower risk of infection because the skin barrier is restored faster, getting back to normal life sooner. It's a huge potential benefit.
Absolutely huge. Did they see improvements in specific aspects of the wounds beyond just the overall score? You mentioned the Bates-Jensen tool has individual items.
Yes, and they analyzed those too. The egg white group showed statistically significant improvements, again, p less than 0.0001 for all of these, across a whole range of wound characteristics compared to the control group.
Like what specifically?
Okay, things like the wound depth decreased faster. The wound edges looked healthier, less undermining, which is where the edge pulls away. There was a quicker reduction in necrotic tissue. That's the dead stuff you need to get rid of. Both the type and the amount improved faster.
That sounds crucial for a clean healing.
Definitely. Also, the exudate, the fluid weeping from the wound, improved less of it and shifting towards healthier types of fluid. The skin surrounding the wound showed better color, less hardening or induration, and less swelling or edema.
Okay.
And perhaps most importantly for actual tissue repair, they saw significantly better formation of granulation tissue, that healthy, red, bumpy tissue that fills the wound, and faster epithelialization, which is the new skin cells growing across the top to close it off.
So it wasn't just like closing faster superficially. It was promoting better quality healing from the base up and across the surface.
That's exactly the picture it paints. A more robust, efficient healing process seems to be stimulated.
Now, you mentioned they also measured wound size. Did that show a difference?
This was an interesting point. While both groups' wounds obviously got smaller over the 15 days, the difference in the rate of size reduction between the egg white group and the control group wasn't actually statistically significant.
That seems odd, given all the other positive results. Any idea why the size measurement wouldn't show a difference?
Yeah, the study authors touch on this. For these kinds of partial thickness burns, the main way they heal is by those new skin cells growing in from the edges and from leftover hair follicles or sweat glands within the burn area that's epithelialization. Big wound contraction, like you see in deeper wounds, isn't the primary mechanism here.
Okay.
So it's possible that while the egg white significantly boosted the quality of the healing tissue underneath and the speed of those skin cells migrating across, which the Bates-Jensen tool picked up, the simple measurement of the surface area didn't reflect that underlying improvement as clearly. Maybe the overall closure rate in terms of square centimeters per day ended up being roughly similar by day 15, even if the way it closed was much healthier in the egg white group.
That makes sense. The detailed assessment tool captures more nuance than just a ruler. What about side effects or complications?
This was another really positive finding for the egg white group. The researchers reported absolutely zero complications in the patients who received the egg white formulation.
None at all.
None reported in the study, period. However, in the control group, the ones getting the standard silver cream plus the placebo ointment, 5 patients actually developed allergic reactions.
To the placebo
No.
or the standard cream?
The study attributes it to the placebo group's regimen, potentially the placebo ointment components or maybe an interaction. But they reported itching, redness, hives. It was significant enough that those 5 patients needed treatment with corticosteroids to manage the reaction.
So the egg white formulation wasn't just seemingly more effective, but also potentially better tolerated or safer in this trial compared to the control setup.
That's certainly what this specific data suggests for this patient group. It points towards a good safety profile alongside the efficacy benefits.
Okay, really impressive results overall. How did the researchers think the egg white was actually working? What are the proposed mechanisms behind these benefits?
They discussed several possibilities based on what we already know about egg white's composition. First off, antimicrobial activity.
Fighting infection.
Exactly. Egg white contains natural compounds like lysozyme, which is famous for breaking down bacterial cell walls, and other proteins like globulins and immunoglobulin Y, which also have roles in immunity. Keeping bacteria under control in a burn wound is absolutely critical. Infection stalls healing dramatically.
Right, makes sense. So infection control is one piece. What else?
They also highlighted specific molecules that could directly promote tissue repair, for instance, tryptophan, an essential amino acid found in egg white.
Tryptophan, isn't that the sleepy stuff in turkey?
Well, it's a precursor to serotonin, which might play a role, but in wound healing, tryptophan itself is thought to help with re-epithelialization, getting those new skin cells to cover the wound. It's also linked to promoting cell proliferation, just making more cells, and neovascularization.
Neovascularization.
Formation of new blood vessels. You absolutely need new blood vessels to bring oxygen, nutrients, growth factors, immune cells, everything the healing tissue needs to rebuild. Without good blood supply, healing just doesn't happen properly.
Got it. So egg white might provide fuel and building blocks and help build the supply lines.
That seems to be the idea. They also mention other components like riboflavin, vitamin B2, and L-arginine. L-arginine is another amino acid, and it's involved in making nitric oxide, which affects blood flow and immune function. Some studies suggest it might help increase growth factor production too.
Okay, lots of potential players. Anything else?
Yes, antioxidants and essential trace elements. Egg white contains vitamins A, E, and C, plus minerals like zinc, copper, selenium, and the antioxidant glutathione. All of these are known to be important for wound healing. They help protect cells from damage, support collagen production, the main structural protein in skin, and generally support the repair processes.
So it's likely not just one thing, but a combination of antimicrobial growth promoting and protective effects working together.
That's what the source suggests. a kind of synergistic effect from all these beneficial components naturally present in egg white.
So if we bring this back to you, the listener,
Yeah.
what's the big takeaway from this first clinical trial, according to the source?
Well, the main implication stemming from that significantly faster healing time, potentially 5-10 days instead of 10-17 days, is the possibility of reduced hospitalization time, although this was outpatient. Faster healing implies less need for intensive care in general.
Right.
It also suggests a lower risk of infection, both because the wound closes faster and because of those potential antimicrobial properties. And logically, if recovery is faster and complications are fewer, it could lead to lower overall healthcare costs. Think about less need for dressings, medications, clinic visits.
That's a really compelling package from a simple natural source. Were there any limitations the authors mentioned about this study?
Yes, they were upfront about that. Being an outpatient study means less control over what patients do between visits. Things like their diet, their general activity, how meticulously they followed wound care instructions at home. These can all influence healing and are harder to control perfectly compared to patients admitted to a hospital.
Sure.
Although they did recommend specific diets and instructions, real life always has variability. But, that said, the strength of the triple-blind, randomized design helps to mitigate those factors and makes the conclusion about the egg white's effectiveness pretty robust, despite those real-world limitations.
Okay, a very strong start, a relatively simple ointment showing clear benefits. Now, let's switch gears quite dramatically. Our next source takes egg white into, well, a much more high-tech direction. We're talking advanced 3D printed hydrogels carrying stem cells for wound dressing. This is from a paper in Frontiers in Bioengineering and Biotechnology.
Yeah, this is a big leap in complexity. This study tackles a different and often much tougher problem: chronic wounds.
Like diabetic ulcers.
Exactly. Diabetic foot ulcers, pressure sores, venous leg ulcers, wounds that just get stuck and refuse to heal properly, sometimes for months or years. They're a huge burden, incredibly costly for healthcare systems, and devastating for patients' quality of life.
And standard dressings often aren't enough for these.
Standard dressings are important for protection, absorbing fluid, maintaining moisture, but they're mostly passive. They don't actively kickstart the healing process in a wound that's biologically stalled. There's a real need for therapies that can actively stimulate repair in these difficult cases.
And that's where stem cells come in.
It's one of the most exciting areas, yes. Specifically, adipose-derived stem cells, ASCs. These are stem cells harvested from fat tissue.
Why those particular stem cells?
A couple of reasons. They're relatively abundant and easy to get. A small liposuction procedure can yield millions of them. But more importantly, they're like little factories releasing a whole cocktail of beneficial molecules, growth factors, cytokines, exosomes.
Signaling molecule.
Exactly. Signals that tell the surrounding cells in the wound, hey, start dividing, build new blood vessels, calm down the inflammation, remodel the tissue. They orchestrate the repair process.
Okay, so ASCs are great, but you can't just sprinkle them on a wound, right? How do you get them there effectively?
That's the critical challenge. Simply injecting them can damage the cells, and even if they survive, they might get washed away by wound fluid, or the healing factors they release might diffuse away too quickly. You need a way to deliver them, protect them, keep them localized at the wound site, and allow them to function over time.
A delivery vehicle.
Precisely. And that's where hydrogels are really useful.
We heard about hydrogels in the first study's ointment base.
Right. But here we're talking about hydrogels as a structural scaffold. They're mostly water held in a 3D polymer network, so they mimic the natural environment of tissues really well. They can provide a moist, protective niche for the cells.
Okay, but this study uses 3D printed hydrogels. What problem does the 3D printing solve compared to just a regular hydrogel?
So the issue with many conventional hydrogels is their internal structure. Often the pores within the gel are very small and maybe not even well connected. Think of like a very dense jello.
Okay.
This tiny pore size really limits the movement of things in and out. Oxygen and nutrients struggle to get in to feed the cells deep inside the hydrogel. And just as importantly, the beneficial factors the stem cells release struggle to get out to the wound tissue, and waste products from the cells struggle to get removed.
So the cells might not function optimally or even survive well inside a dense hydrogel?
Exactly. It can stifle them. This is where 3D printing offers a huge advantage. It allows you to design and build the hydrogel scaffold layer by layer with incredible precision. You can create a structure with much larger interconnected pores, like a well-designed scaffolding or lattice.
Like building little tunnels and channels throughout the gel.
Precisely. Imagine creating an internal highway system within the hydrogel.
What are the benefits of that controlled, porous structure?
Several key things. First, much better transport. Oxygen, nutrients, growth factors, waste products, they can all move much more freely. This keeps the embedded stem cells healthier and allows their secreted factors to reach the target tissue effectively. Second, the defined structure can potentially provide physical cues to guide how the new tissue grows and organizes itself within the wound. And third, 3D printing allows for better control over the overall shape, size, and mechanical stability of the dressing.
Okay, so 3D printing allows for a smarter, more functional scaffold and the material they chose to print with.
Back to our star ingredient, egg white. Again, they highlight its low cost, abundance, and the fact that it's a natural protein source. But here, they're leveraging its potential as a biocompatible material that contains molecules that might inherently support cell growth, making it a potentially bioactive ink for their 3D printer.
Wow. From ointment to bio-ink. How did they actually make this 3D printed egg white dressing? Sounds complicated.
It is a multi-step biofabrication process. They started with just fresh, liquid egg white. Then, to make it printable, they needed to turn it into a gel. They did this using a chemical cross-linking method involving a brief treatment with sodium hydroxide, NaOH. This causes the protein molecules to link together, forming a printable hydrogel.
Okay, so step 1, make egg white gel. Step 2, print it.
Exactly. This egg white hydrogel was then loaded into a specialized 3D bioprinter. The printer carefully extruded the gel, following a pre-designed pattern, to build up a mesh-like scaffold structure. The paper mentions dimensions of about 2.5 by 2.5 centimeters and only 0.2 centimeters thick, quite a thin mesh.
And was that it? Just print and use?
Not quite. There was a crucial final step. After printing, these delicate egg white mesh structures were soaked in a standard cell culture medium called DM. This soaking did two things. It neutralized any remaining NaOH, making the scaffold safe for cells. Very important. And second, it induced a secondary cross-linking, basically strengthening the structure, making the hydrogel more stable and robust. This final, stabilized 3D printed structure was what they called 3D SEWH.
3D. Sodium hydroxide cross-linked egg white hydrogel.
Got it. And then they added the stem cells, the hitching part.
Right. Once they had these optimized 3D SEWH scaffolds, they seeded them with the adipose-derived stem cells, ASCs.
Yeah.
The idea is the ASCs would attach to and live within the porous structure of the egg white scaffold. So you end up with a living dressing. The scaffold provides the home, the cells provide the therapy.
Okay, a very sophisticated construct. How do they test if it actually worked? Did the scaffold support the cells properly?
They did a whole series of tests. First, they look at the structure using scanning electron microscopy, SEM. Those high-res images confirmed that the 3D printing process was successful. The scaffolds had those designed, interconnected micropores they were aiming for.
Okay, the structure looked right. What about how it behaved physically? Could it hold its shape? Was it suitable for printing? They tested its rheological properties, basically, how it flows and deforms. The results showed it behaved as a viscoelastic gel, meaning it has both liquid-like and solid-like properties. And crucially, it showed shear thinning behavior.
Shear thinning. We heard that term before. Good for printing.
Yes, exactly. It means the gel becomes less viscous, flows more easily when you put pressure on it, like when it's being pushed through the fine nozzle of the 3D printer. But as soon as that pressure is released, it thickens up again and holds the printed shape. It's ideal for extrusion-based 3D printing. They did mention a minor technical issue called wall slip in their measurements, which can happen with gels, but overall, the properties were good for fabrication.
Right. Structure good, printing properties good. What about biology? Was it safe for the cells? Did the cells like living on it?
This was the critical biocompatibility testing done in vitro in lab dishes. They needed to make sure the 3D SEWH itself wasn't toxic. They exposed cells to the material and also to liquid that the material had been soaked in. The results were really positive. Cell viability remained high, over 84% after 3 days, which was similar to control conditions without the material, so no significant toxicity.
Right.
And they also looked under the microscope and saw that the ASCs could successfully attach to the scaffold's grid structure and, importantly, proliferate, meaning they could multiply and spread out over the surface. The egg white scaffold was providing a hospitable environment.
Okay, so the cell survived and grew on the scaffold. Did having the cells on the scaffold actually boost any healing-related functions?
Yes, They tested this by looking at fibroblasts. Fibroblasts are key players in wound healing. They're the cells that build the new tissue matrix, mainly collagen. They collected the liquid medium that the ASCs had been growing in while on the 3D SCWH scaffold. When they added this conditioned medium to fibroblasts, it significantly boosted the fibroblasts' proliferation rate.
More than just the scaffold alone, or the cells alone.
Yes, significantly more than medium from the scaffold without cells, or from ASCs cultured without the scaffold, or just plain medium. This points strongly towards a synergistic effect.
Meaning the combination is better than the sum of its parts.
Exactly. It suggests the egg white scaffold isn't just a passive carrier, it's interacting with the ASCs, maybe helping them survive better or stimulating them to release more potent healing factors, leading to a stronger pro-healing signal for other cells like fibroblasts.
That's very promising in vitro. Now, for the big test, did this high-tech egg white stem cell dressing actually work in a real wound? What happened in the animal model?
Right, the in vivo study. They used mice, creating standardized, full-thickness wounds on their backs. These are serious wounds that go right through the skin layers.
Oh.
They divided the mice into 4 groups for treatment. A control group, just a standard bandage. A group getting just the 3D SCWH hydrogel scaffold, no cells. A group getting just the ASCs applied directly to the wound, no scaffold. And the key group getting the combined 3D SCWH plus ASCs dressing.
And what did they see? Which group healed best?
The results were very clear. The group treated with the combined 3D SCWH plus ASCs dressing consistently showed the fastest and most complete wound closure over the study period.
How much faster? Do you have numbers?
Yes. By day 14, which is pretty quick for this type of wound model, the wounds in the combined treatment group were nearly healed. Only about 2.1% of the original wound area was still open.
2%, that's almost closed. How did the others compare at day 14?
Much worse. The control group wounds were still about 16.2% open. The group with just the egg white scaffold, 3D SCWH alone, was about 10% open.
Oh.
And even the group with the stem cells, ASCs alone, was still about 6.5% open.
Wow, so the combination really outperformed everything else, including the cells or the scaffold on their own.
Dramatically so. The difference started becoming statistically significant around day 7, and then the combined group just pulled further and further ahead, especially in that second week, which is crucial for tissue formation and closure.
Did they look inside the healing tissue to see why it was healing better in that group? What mechanisms were at play?
Yes, they did histological analysis looking at tissue slices under the microscope with special stains. Using H&E and Masson's trichrome stains, which show tissue structure and collagen, they observed that the tissue from the 3D SCWH plus ASC group had a significantly higher density of newly formed blood vessels. That's angiogenesis again. They also saw more organized collagen deposition and a thicker, more developed new epidermal layer covering the wound.
So better blood supply, better structural rebuilding.
Exactly. And they backed this up by looking for specific protein markers using immunohistochemistry. They found higher levels of VEGF, that's vascular endothelial growth factor, a key driver of blood vessel growth.
Consistent with the increased vessels they saw.
Right. And they also found higher levels of vimentin, which is a marker associated with active fibroblasts. This supports the earlier in vitro finding that the combination stimulates fibroblast activity.
So putting it all together, the 3D printed egg white scaffold provides a good home, maybe even actively helps the ASCs, which then release factors that strongly promote new blood vessel growth and fibroblast activity, leading to faster, better quality tissue regeneration.
That's the mechanism strongly suggested by all the data presented in the source. It's a powerful combination therapy.
So connecting this back to you, the listener, think about what this could mean for treating those really tough chronic wounds. We're moving beyond just passive dressings. This is an engineered, active biological treatment.
It really is. Using a cheap, natural material like egg white, but shaping it with advanced 3D printing and then loading it with therapeutic stem cells. It's a glimpse into the future of personalized wound care.
It could potentially mean faster healing, fewer infections, maybe even better scar outcomes for wounds that currently just don't respond well to treatment. It really builds on the potential seen in the first study, but takes it to a whole new level.
A much more sophisticated application tackling a harder problem.
Absolutely amazing trajectory. Okay, so wounds heal faster, maybe even chronic wounds respond better, but healing isn't always the end of the story, right?
Yeah.
Sometimes there are lingering issues like changes in skin color. Our third source looks at this, how egg white might play a role in managing skin pigmentation after wounding. This paper is from the journal Molecules.
Yes, this one focuses on something called post-inflammatory hyperpigmentation, or PIH. It's really common, especially after burns, acne, eczema, even just scratches or cosmetic procedures for some people. You get these dark patches or spots where the inflammation or injury was.
And it's more noticeable in some people.
Yes, it tends to be more pronounced and persistent in individuals with darker skin types, although it can affect anyone. And it can be quite distressing cosmetically, sometimes lasting for months or even years.
And fundamentally, PIH is caused by too much pigment.
Exactly. Too much melanin. The inflammation or injury triggers the skin's pigment-producing cells, the melanocytes, to go into overdrive. They make excess melanin and deposit it in the surrounding skin cells.
So if you want to prevent or treat PIH, you need to slow down melanin production.
That's the main strategy. And the key enzyme that controls the rate of melanin production is called tyrosinase. If you can inhibit tyrosinase, you can usually reduce melanin synthesis.
Tyrosinase inhibition is the goal, and this study connects egg white to that.
It does. The source mentions the traditional use of egg white in skincare and cosmetics, but also points to prior scientific work. Apparently, earlier research found that if you break down egg white proteins into smaller pieces, called peptides, some of those peptides can inhibit tyrosinase activity, at least in simple test tube assays using mushroom tyrosinase.
Mushroom tyrosinase. Okay, maybe not exactly human skin, but a starting point.
Right. And they specifically mention one peptide sequence, GYSLG-NWVC-ACO, that was identified in previous work as being quite effective against that mushroom enzyme.
So this current study aimed to take it further. Look at human-relevant cells.
Precisely. The goal here was to see if that specific peptide, GYSLG-NWVC-ACO, and also different mixtures of peptides from broken down egg white, they called these hydrolysates, CEWH mono and CEWH dia, could actually inhibit tyrosinase activity and reduce melanin production inside living cells. And to try and figure out how they were doing it, the mechanisms involved.
What kind of cells did they use for this?
They used a common cell line in pigmentation research called B16F10 melanoma cells.
Melanoma, cancer cells.
Yes, they're mouse skin cancer cells. But the reason they're used so widely is that they actively produce melanin, much like human melanocytes do, and they use very similar enzymes and signaling pathways. So they provide a reliable and convenient model system to test potential effects on pigmentation in vitro.
Okay, a standard lab model. What experiments did they run on these cells using the egg white peptide and hydrolysates?
They did quite a comprehensive set of tests. First, basic safety. They checked for cytotoxicity to make sure the treatments weren't just killing the cells, because dead cells don't make melanin. They found low toxicity at the doses used.
Good. Then the key tests.
Then they measured intracellular tyrosinase activity directly within the cells, specifically its dephenolase function, which is a key step. They also measured the actual amount of melanin pigment the cells produced. To explore mechanisms, they looked at intracellular levels of cyclic AMP, or AMP, that's a signaling molecule that often boosts melanin production.
Okay, enzyme activity, melanin levels, signaling molecule, anything else?
Yes, they also looked at gene expression. Using a technique called RT-qPCR, they measured the levels of messenger RNA, mRNA, for key genes involved in making melanin, MIF, master regulator, TIR, the gene for tyrosinase itself, and TRPA1 and TRPA2, genes for other related enzymes. Lower mRNA levels generally mean the cell is making less of that protein.
Very thorough. And they compared the egg white stuff to a known inhibitor.
Yes, they used EGCG, a compound from green tea known to reduce pigmentation, as a positive control.
Okay, so what were the main findings? Did the egg white components work? Did they reduce pigment?
Yes, they did, significantly. Both the specific peptide and the hydrolysates managed to reduce both the tyrosinase activity inside the cells and the overall amount of melanin produced. The effects were quite strong, particularly at a concentration they tested of 10 micrograms per milliliter.
How strong? Can you give us some figures?
Sure. At that 10 gml dose, the peptide GYSLG-NWVC inhibited tyrosinase activity by about 45%. But interestingly, it inhibited the actual melanin level by over 70%.
70% reduction in melanin. That's substantial.
It is. And the paper notes this melanin inhibition was actually better than what they saw with the EGCG control at the same dose.
Wow. What about the hydrolysates?
They also worked well. One called CEWHD inhibited tyrosinase by about 51% and melanin by about 48% at 10 gml. The other, CEWH mono, didn't inhibit the enzyme activity as much at that dose, but still strongly inhibited melanin levels by almost 70%, similar to the peptide.
Okay, so clearly these egg white derivatives have anti-pigmentation effects in this cell model. Now, you mentioned something about a puzzling dose effect with the peptide.
Yes, this was a really intriguing finding. Normally, you'd expect a stronger effect with a higher dose, but for the peptide, they found that when they increased the concentration above 10 gml, the effect on reducing melanin actually started to decrease a bit. It wasn't a simple dose-response curve.
That is weird. More peptide led to less melanin reduction. Why would that happen?
The researchers proposed a clever hypothesis based on pH. They measured the pH of their test solutions and found the peptide and hydrolysates made the solutions slightly alkaline, or basic pH, between about 7.9 and 8.8.
Okay, and how does that relate?
Well, tyrosinase, the enzyme, actually works best at a slightly acidic to neutral pH, around 6.5 to 7.0. So making the environment more basic would naturally slow the enzyme down a bit. That fits with the tyrosinase inhibition they observed.
Right, basic pH inhibits the enzyme, but that still doesn't explain why more basic, at higher peptide concentration, would lead to more melanin overall.
Exactly. That's the second part. Melanin formation isn't just the enzyme step. There's a later chemical process called polymerization, where the melanin precursor molecules link together to form the final dark pigment. The authors suggest that a basic environment, maybe particularly the basic amino acids like lysine in the peptide sequence, might actually promote or speed up this polymerization step.
Ah, so the peptide might be doing two opposing things. Inhibiting the initial enzyme because of the basic pH, but also potentially speeding up the final pigment formation step because of the basic pH.
That's exactly the hypothesis. At low concentrations, the enzyme inhibition might dominate. But at higher concentrations, the propolymerization effect might start to catch up, partially counteracting the enzyme inhibition, leading to a less than expected overall reduction in the final melanin pigment.
That's fascinating, a really complex interplay. It shows how just changing the concentration of something can have nonlinear effects if it influences multiple steps in a pathway differently.
Absolutely. It highlights the need to look beyond just simple enzyme inhibition sometimes.
What about the signaling pathway they investigated, 10-AMP? Did the egg white components affect that?
Yes, they found that the peptide, GY-SLG and WVC-CSAG, and one of the hydrolysates, CEDWHD, both significantly reduced the levels of intracellular gamma-P by about 13–14%. This was similar to the effect of the EGCG control.
And lower CAMP usually means less melanin signaling?
Generally, yes. High CAMP often stimulates melanin production. So this suggests that part of how the peptide and CEWH work might be by dampening down this pro-pigmentary signaling pathway. Interestingly, though, the other hydrolysate, CEWH mono, did not significantly change CAMP levels, even though it strongly reduced melanin.
So CEWH mono must be working through a different pathway altogether.
It seems so. It implies there are multiple ways these different egg white components might be interfering with the pigmentation process at the signaling level.
Okay, and what about the final piece, the gene expression? How did the egg white components affect the cell's instructions for making melanin-related proteins?
This also showed some interesting differences. The two hydrolysates, CEWH mono and CEWHD, behaved in a way that's more commonly seen with anti-pigmentation agents. They both reduced the mRNA levels of all the key genes tested: MIF, TIR, TRPP1, and TRPP2.
So turning down the instructions for the whole pathway.
Essentially, yes. Less mRNA usually leads to less protein being made, so less enzyme activity, less melanin. For CEWHD, this could be linked to the CAMP reduction. For CEWH mono, which didn't affect TAMP, it must be acting via other signaling pathways that regulate these genes. The paper mentions possibilities like PKA, PI3K ACT, MPK, or WANT pathways, which are all known to influence pigmentation.
Okay, so the hydrolysates seem to be shutting down production signals. What about the specific peptide, GY-SLG and WVC-SAG? Did it do the same?
No, this is where it got really intriguing again. The peptide did reduce the mRNA for tyrosinase TYR, which fits with it reducing the enzyme's activity.
Yeah.
But counterintuitively, it actually increased the mRNA levels for TRPP1 and TRPP2, the other enzymes in the pathway.
It increased them.
Yeah.
That makes no sense if it's trying to reduce melanin.
Right. And it also had no significant effect on the mRNA for MIF, the supposed master regulator. This pattern is quite unusual and doesn't align with simply switching off the melanin production genes.
So if it's not consistently turning down the genes, how did it manage to reduce melanin levels by 70%?
This led the researchers to propose another layer to its mechanism: action at the post-translational level.
Post-translational, meaning after the protein is made.
Exactly. Gene expression, mRNA, is about making the blueprint. Translation is building the protein from the blueprint. Post-translational is anything that happens to the protein after it's built. Maybe it gets modified, activated, inactivated, or even targeted for destruction.
Ah,
So the hypothesis is that the peptide might be working primarily by interacting with the tyrosinase protein after it's made, perhaps directly inhibiting its function, or maybe causing the cell to degrade the tyrosinase protein, or even the MITF protein faster, even if the mRNA instructions are still around.
so it could be like sabotaging the workers or throwing away the finished product rather than just shutting down the assembly line instructions.
That's a good analogy. It would explain how you can get a strong reduction in enzyme activity and melanin levels, even with those confusing gene expression results. It suggests a more direct interference with the key players after they've been produced.
Okay, that adds yet another layer of complexity to how this simple-sounding peptide might be working. So let's bring this third study home for the listener. What's the implication here?
Well, think about the whole healing journey we've discussed. Study 1 suggested egg white could help wounds heal faster. Study 2 showed it could be part of advanced dressings for tough wounds. This third study now suggests that components derived from egg white might also help manage one of the common aftermaths of healing, that unwanted darkening, the PIH.
So it could potentially offer benefits across multiple stages of wound recovery.
Potentially, yes. Helping the initial healing, maybe supporting complex wound closure, and then potentially mitigating the cosmetic issue of hyperpigmentation afterwards. That would be particularly relevant for people prone to developing PIH. It's another example of finding sophisticated biochemical activity in a very common natural source.
It really ties things together. It seems egg white's potential is surprisingly diverse, antimicrobial, helping tissue grow, providing structure for advanced dressings, and even influencing pigmentation biochemistry.
Absolutely. And the mechanisms are clearly complex, involving multiple signaling pathways like CAMP, changes in gene expression, potential post-translational effects, and even basic physical properties like pH playing a role. It's not just one simple action.
What an incredible deep dive this has been.
Hmm.
We really traced egg white from, something akin to a traditional remedy in that first burn study,
Right, a simple ointment.
then saw it become a sophisticated component in cutting-edge biomedical engineering with those 3D printed stem cell-loaded scaffolds.
A huge technological leap.
And finally, explored how its specific peptides might target skin pigmentation issues at a really detailed cellular and molecular level.
It's fascinating to see science investigate a common food ingredient in such different ways, tackling distinct challenges within the whole arc of wound healing and recovery, from basic repair to complex regeneration to aesthetic outcomes.
It really makes you think, doesn't it? Considering these studies, what other everyday natural materials, maybe things used in traditional medicine for centuries, are just sitting there holding untapped potential for serious biomedical uses?
That's the provocative question, isn't it? How much more is there to discover by applying modern scientific rigor to understand the basis of traditional practices, or simply exploring the biochemistry of common natural products?
It feels like there could be a lot more hidden gems out there.
I suspect you're right. The intersection of nature, tradition, and cutting-edge science is likely to keep revealing surprises.
Well, thank you for joining us on this fascinating exploration of the science locked inside egg white. As always, we encourage you to keep exploring, keep asking questions, and notice the science that's often hidden in plain sight in the world around you.